Hi, my name is Erika Muhlrad, and I am a sophomore here at the University of Utah studying biomedical engineering. And I've been working with Dr. Amy Lenz in the Orthopedic Research Lab here at the U. This project, Standardized Coordinate Definitions for Foot and Ankle Biplane Fluoroscopy, is an IRB approved protocol and consists of 27 individuals. In the Lens group at the Orthopedic Research Lab, we use an imaging technique called biplane fluoroscopy or biplane fluoro. Biplane fluoro uses two X-ray video cameras running simultaneously and capturing two different views of a subject performing a task like standing or walking. The setup for biplane fluoro is shown in figure 2. A separate CT scan is made into a 3D computer model of each of the bones in the foot, called a digitally reconstructed radiograph or DRR. By lining up the 3D bone models to the two views of the X-ray from the biplane fluoro, we're able to precisely calculate and quantify motion of each of the small bones in the foot and ankle. The precision with biplane fluoro is something that's not possible with skin-based motion capture techniques. After different frames of the fluoroscopy data are aligned with the DRR, a coordinate system is assigned to each bone. After a thorough literature review, it showed that many research studies that use biplane fluoro do not use the same coordinate systems to calculate bone kinematics in the foot and ankle. This makes it really difficult to compare findings between studies and across different populations. This study's objective is to compare different coordinate systems used in previous literature and determine and analyze the effectiveness and reliability of each. Finally, we want to propose a single standardized coordinate system for foot and ankle biplane fluoroscopy kinematics. One widely used coordinate system is the principal component axes, or PCA. These axes can be computer generated, and there is zero error from user input. Although easy to apply and error-free, principal component axes depend solely on the shape of the bone. This means for different populations of bones, such as those with osteoarthritis, may be drastically different and hard to compare to a healthy bone set. Aside from this, PCA do not always align with meaningful parts of the bone, like the articular joint surfaces. We want to assure that both engineers and clinicians are able to value the data from the axes applied to the bone models. In addition to the meaningfulness, other aspects of the bone models must be considered when choosing a coordinate system. In figure 4, you can see that the PCA applied to the tibias are very scattered, unlike the tight clusters on the talus and calcaneus. This is to do with the short amount of tibia shaft that was imaged. This specific challenge will be investigated further to determine the amount of tibia needed to create consistent and valuable axes. Other than principal component axes, in the future, we'll compare axes generated from various bony landmarks and geometric shapes fit to the bone. Finally, we want to determine a standard and meaningful coordinate system to be used across the field of foot and ankle biomechanics biplane fluoroscopy studies. I'd like to acknowledge the funding source for this project, Shriners Hospital for Children, and thank you for taking the time today to view my work.